Under the sterile, fluorescent glow of a laboratory sequencer, a routine maternity blood test is processed to confirm a biological link. But the results trigger a genetic impossibility. The test indicates that the mother who just delivered her newborn baby is not biologically related to the child. This glaring discrepancy creates a medical and forensic nightmare. Because the laboratory profile fails to match the child, the hospital records are overruled by the genetic data, leading the law to treat the mother as a stranger to her own biological offspring. This chart illustrates the conflict. A buccal swab from the epithelium of her inner cheek reveals one genetic profile, while the mitochondrial DNA found deep within her ovarian tissue tells an entirely different story. Her body is secretly harboring the genetic ghost of a twin absorbed before she was born, a hidden biological mystery that only surfaces when the blood and the bone disagree. To find the origin of this anomaly, we must look back to the dark, fluid environments of the early womb. Two separate zygotes, each carrying an entirely unique genome, drift slowly through the dark amniotic fluid and collide. Instead of developing into fraternal twins, the cellular membranes of these distinct embryos dissolve, causing them to physically merge into a single cluster of stem cells. This creates a tetragametic chimera, one person built from two entirely different genetic blueprints. As the fetus develops, these competing stem cell lines migrate across the embryonic disc. A liver might expand using the DNA of twin A, while the cardiac muscle of the heart is built entirely from the blueprint of twin B. Sometimes the secret becomes visible on the surface, appearing as a sharp line of dermal pigmentation, or Blaschko's lines, that swirl across the skin and glow under ultraviolet light. Early development functions as a complex construction site, where billions of alien cells must negotiate a delicate peace treaty to build a single functioning organism. This raises a question. If the womb is chaotic enough to fuse two people together, how does it manage to construct an organ as precise as the human eye? The process begins at the pulsating neural tube, the starting point for the visual system. Optic vesicles bulge laterally, pinching off to form two distinct pockets. Internal pressure drives these vesicles outward, using mechanical force to carve space for growth. This expansion is the mandatory first catalyst in a continuous chain reaction of cellular origami. The advancing optic vesicles eventually reach the outer boundary of the embryo, meeting a layer called the surface ectoderm. When the vesicle strikes the rigid surface ectoderm, the physical impact causes the tissue to buckle under the stress. This collision triggers an inward structural collapse, forcing the vesicle to fold back on itself. This double-walled structure is the optic cup, the foundational shape of the eyeball. This inward fold provides surface area to weave the complex, multi-layered retina. In this context, the structural collapse is not a failure, but a mechanical necessity required to create the sensory surface of the eye. The newly formed optic cup immediately creates heavy logistical demands, requiring a constant flow of resources to sustain its growth. The hyaloid artery acts as the vital lifeline, penetrating the center of the structure to feed the rapidly dividing retinal cells. Simultaneously, this sensory organ must be wired back to the brain. Embryonic tissue bundles along the stalk to form the thick cable of the optic nerve, creating a direct data path from the eye to the brain. The simultaneous execution of vascular feeding and neural wiring highlights the high level of coordination involved in fetal tissue mechanics. Pulling back from the microscopic folding process reveals the fully formed, intricately woven human optical system. This hyper-refined sensory organ begins as a simple outward pinch of the neural tube. Whether a body is built from a single genetic blueprint or two competing sets of DNA, it relies on this exact sequence of collision and precise folding. This microscopic ballet of folding and wiring dictates how we perceive the world around us. It is through these chaotic biological collisions in the early womb that the structural precision of human sight is able to emerge.